Okay, this video is about chapter 12, Pharmaceuticals and the Economics of Innovation. For this chapter, we're going to introduce the history of pharmaceutical industry, and we're going to talk about induced innovation and the economics behind it. So the pharmaceutical industry got its start in 1899. When Bayer, which is a very famous company until today, a German chemical company introduced a painkiller called aspirin. Today, the pharmaceutical industry is massive but tightly regulated. This industry is an ideal setting to study both the economics of innovation and the economics of regulation. Okay, so a life cycle of a drug is first, you have to find a chemical compound that might treat the disease. There is trying, there is error, and there is rational drug design. So for example, US pharmaceutical industry in 1970 tested over 700,000 substances in animals, of which only about 1,000 were promising enough to pass into human trials. So once you find a chemical, you test it on animals to show whether it is not toxic. And then you test it on humans in 3 different phases. In phase 1 and phase 2, it's usually about safety. So low dose to healthy individuals, and then dose to unhealthy individuals. Phase 3 is usually the largest scale, or sometimes the final stage that you test effectiveness in preventing disease or medical conditions. This takes about 2 years for phase 1 and phase 2, and then 3 to 4 years in phase 3. Once you finish your phase 3, you get the data, you do the statistical analysis. It is statistically significant, and it is promising enough, so then you can apply to FDA and get approved for sale by FDA or a similar body. Okay, so then once a drug is approved for sale, the drug company that developed this drug has a temporal legal monopoly protected by a patent. In US, this is 17 years. This is the company's chance to recoup the millions of dollars spent on testing and developing. And after that time is up, other companies can produce the same drug cheaply and profits decrease sharply. Okay. Drug development is very costly. It is very hard to first find the chemical in the first place. And after you find that chemical, you have to pass 3 phases of trial, and only 21.5% of the drugs that entered phase 1 pass all the way to phase 3. And this whole process can cost up to 500 million. I believe nowadays it could be even more costly to bring a drug to the point of approval. In this 2011 paper, it estimates the average cost of bringing a drug to market between 1970 to 2000 range between 92 million to 800 million. And while these estimates do vary greatly, the consensus is that the cost of developing new drugs have risen dramatically over time, because it is harder and harder to make technology innovation when all the easier innovations are already done. This is a graph of success rate by phase. You can see that in phase 1, only 71% of them success. And then phase 2, phase 3, all the way, only 21.5% survived. Okay, so then once we have approved a new drug, the company that developed it is gonna get patent. Patent is a government sanctioned, it's a government protected, or it's a law protected monopoly in a limited market, usually for one specific idea, algorithm, or products. Patents are typically awarded to the first person or company that invents a new technology. So because there's only one person or one firm, so this is a monopoly. This is a legal monopoly, and hence the opportunity for monopoly profits because you are the only one that supplies it. So monopoler has the ability to set price in the price that maximize their profits. In practice, only the top 30% of drugs pay for themselves. What does it mean by pay for themselves? That means that the profits, or in this graph, the revenues from drug sales are able to cover their R&D cost or have something to spare. So then that comes the question, if some drug that cannot end up paying for their own cost, who is paying? Right. So pharmaceutical R&D expenditures would have fallen 64% without patent protection, compared to just 8% in other industry. I think the question, the answer to this question is that some companies, they hedge this risk by inventing many drugs. So then they can subsidize their not making profit drugs from their profit-making drugs. And some of them, of course, it's subsidized by the government, and therefore it comes from tax incomes. Okay. In terms of the economics of patent, there is a trade-off between a stronger and weaker patents. The downside of a strong patent is that customers will have to pay monopoly prices for a longer period. So for example, 17 years is long for some drugs, but in some countries, maybe it takes 10 years. Then in that circumstances, then customers will be able to get cheaper drugs after 10 years of monopoly price, because the other companies can copy this innovation and drive the price down through competition. There will be less incentive for further innovation by the same company, because if this patent is already making this much money, then they have less motivation to develop another one. There would also be legal barriers for subsequent innovations by another company. This for example, if the other company wants to use a part of this company's new drugs that is protected by its patent, then it will be costlier to buy that patent to develop a subsequent ones. Okay, so that's the downside of patent strength, but the good side is that if the patent is too weak, then there is no incentive to develop a new drug in the first place. So we need patent, and we want the strength of patent to be at a level that maximizes the society welfare. In developing countries, low-income countries think about this trade-off differently. So monopoly prices weigh more heavily on low-income populations. There's also a free rider effect, that if the US has patent protections, and companies will develop new drugs, even if there are weak patent protections in some other countries like India. We will see this effect again when we talk about price controls. There's also the economics of price discrimination. In theory, drug companies could sell their drugs for different prices in different countries. So here, price discrimination means that you have the same products, but different type of customers gets a different price tags. In practice, black market is part of the price discrimination. Black market importation makes this impossible. Price controls, price ceilings are usually set or negotiated by the government. So for example, in Italy, government publishes a list of maximum permissible prices for each drug. You can think of monopoly firms will set a price that is at a level that maximizes their profit. And if this level is higher than a level that government thinks will hurt customers, then the government can cap its maximum price. For example, in UK, NHS, their National Health Department, sets the price at which they are willing to purchase drugs. In this case, because there is one buyer in this whole country, so the power shifts from the firm to the buyer, and the buyer came down with a negotiation power. We call that monopoly power. Okay, price controls hurts the company and therefore hurts incentive for research. There's a trade-off between the access to existing drugs and incentives to develop new ones. This is the same as patent trade-off. US have no broad price controls so far, while other countries free ride on US markets for future innovation. Drug companies count on making their money off the US consumers. Okay, so then the next part, we're going to talk about induced innovation. So this part of the innovation is induced by usually a financial incentive. Definition, induced innovation means that discoveries that results when innovators change their research agenda in response to profit opportunities. Okay, so we think of innovators in the medical field. A lot of them might be motivated by wanting to save more people's life, wanting to change people's health, wanting to make people live longer and happier. But a lot of them, as long as they need funding, right, then they need a certain level of profit opportunity. For example, we have a changing demographics in the United States. In the past several decades, the US population has aged a lot. We have an older and older population. So then drug companies know that this aging population and this aging population that has a certain amount of wealth to pay for their healthcare and have a higher demand for their healthcare will make bigger markets. So drug companies turn their attention to drugs for the elderlies. Academic and public institutions also engage in induced innovation, even though they do not usually profit directly from their discoveries. So for example, large-scale production of penicillin during World War II by the US Department of Agriculture. In recent years, academic researchers have focused more on obesity. US Army ceasing malaria research after Vietnam War ended and troops came home from the malarial regions. So these are research that can be done, or we don't lack any scientists or resources to do it, but we don't have a demand or we don't have a profit incentives for them. So the research agenda changes. Is this good? Is induced innovation good or bad? Who is harmed? We can easily think of places where people cannot afford new drugs will be harmed by this type of induced innovations because they don't provide financial incentives. So for example, diseases that are rare, like orphan disease, or that mostly occurred in developing countries, or just a subpopulation that have less wealth or have a smaller size of population, they will receive less attention from researchers because there are less profit to be made. Governments should come in to help. There are governments that have tried to harness the power of induced innovation to fight this disease. So in US, there is Orphan Drug Act. And there's also advanced purchase of yet undiscovered vaccines for HIV, malaria, or TB. Next, we're going to talk about regulations of the pharmaceutical industry. So we heard about FDA a lot, especially during the pandemic. FDA regulations came in to ensure that all the drugs that come into the market are safe for people to use. So before 1930s, there weren't any safety or testing regulations for the drugs in the United States. In Europe, 1960, a drug is prescribed to pregnant women with morning sickness, and it caused birth defects in over 10,000 newborns. So this drug is eventually pulled from shelves and promoted stricter drug regulations throughout the world. So then that is the motivation behind the 1962 Falver-Harris Amendment in the US, that they believe that companies must prove that every new drug are safe and efficacious throughout their clinical trials. And in 1973, there is stricter regulations lead to a lower number of newer chemical entities on the market. Okay, so let's talk about how FDA actually looked at when companies submit their clinical trials data. So FDA has to decide how strict they want to be. So for example, FDA want the drugs to be safe, but if this drug tested on 10,000 people and only one of them died, is this safe, or this is not safe enough and you want to reject it? Okay, if you think this is not safe enough, then maybe it's too difficult to find anything that it's 100% safe. So then do you think 99% safe is okay, or 98% safe is okay, right? So that's a dilemma for the FDA. You have to weigh risk and benefits. So a nice framework is called type 1 and type 2 errors. A type 1 error means that you have a bad drug and you approved it. Okay, this is a bad thing to do. And things that happens in that way because a bad drug came in and did all of its clinical trials, and it just happened that they had a very good results. Okay, so then you approved it and it went on market. It doesn't seem to do as well in the market or in general population than in the trials. This results in a bad drug being approved. We call that type 1 error. In the case of vaccine, we can think of a vaccine that is not effective, but is being approved. Okay. For type 2 error, it's the opposite. Good drug is rejected or delayed. For example, beta blockers. In the case of a vaccine, you can think of an effective vaccine, but its clinical trials was probably unlucky, so that they have some people who have severe side effects, or some people who die during the trials. In this situation, if this vaccine or this drug is not approved, but in general public, it might not cause that much death or side effects, then we end up having another situation which we are too careful and we end up rejecting a good drug or good vaccine that could save people's life. Okay, so we have to understand that phase 3 trials do not have complete information about drug, because in any trial, you're only trying it on a limited population. You're not trying it on the whole population. There is no way you can do it. So there is always a problem in statistics, which we call that you only have a sample, you don't have a population, and you have to infer information from this sample to a population. So a nice framework here is type 1 and type 2 errors. Okay, so let's take a look at this graph. This graph is actually a distribution of two types of drugs. And the y-axis is density. Think of it as distribution. X-axis is T. Let's think of it as performance, okay? So of course, a good drugs have good performance, a bad drug have bad performance. And performance is not a fixed value. Performance is a normally distributed variable. You might ask, why is that? Shouldn't a drug have one specific effect? The answer is that because one drug can have different effects on different people. In the case of COVID vaccine, we know that if we test people's blood after they get the vaccine, some people are going to generate more antibodies and some people are going to generate less antibodies. And immune compromised people are going to generate least antibodies, right? So if you think of the amount of antibodies is this x-axis, which is the performance. So here I'm not assuming that antibodies is how effective a vaccine is, but think of it as one of the performance variable that we're interested in. So then good drugs have a distribution that is better, and bad drugs has a performance distribution that's worse. So we can see that these two distributions is not the same. The good drug distribution is in the right side of the bad drug distribution. Okay, but the problem is that they overlap. So think of a bad vaccine. Maybe some people who are really healthy, they can still generate a certain amount of antibodies. And then if you compare this person's blood test with an immunocompromised person who received a good vaccine. So think of a healthy person's blood with bad vaccine and an immunocompromised person with a good vaccine. Then maybe their antibodies amount are the same, or maybe the performance, any performance measured on these two people are the same, or even the one with the bad vaccine are going to have a higher performance than the one with the good vaccine. So we are looking at two distributions that one is better than the other for sure, but they also overlap. So then that creates the difficulty for FDA. That is, you have to draw a line. Let's say in this graph is T star, that if the phase 3 trial data is better than T star, then I think this is a distribution of a good vaccine or a good drug. So I'm going to approve it. But if you come back with a result that's smaller than T star, then I'm going to reject it. And when you reject or approve at T star, the type 1 error is this small gray area on the right-hand side of T star. Okay, so this is the possibility that you approved a bad drug. On the other hand, type 2 error is on the left-hand side of T star. This is the probability that you approved. This is the probability that you had a good drug, but you rejected it. So that's a type 2 error, which is a good drug is rejected. Okay, so how do we actually draw this line? Where should I put the T star in? Then it comes to our next graph. That is, suppose we have 5 different possible thresholds. Let's pick 5 different T stars, T1 star, T2 star, all the way to T5 star. So if you look at T1 star, here we have a very low type 2 error, which is at this point, if we only approve good drugs at this point, then we will not reject any good drugs. Okay, because the distribution of a good drug is completely at the right-hand side of T1 star. But if we reject bad drug at below this point, then we have this, let me probably use, do I have a pen? Yes. Okay, let me highlight. If we approve any drug on the right-hand side of this T1 star line, then now if you focus on the distribution of bad drugs, then this yellow area is the size of the type 1 error that we created, which is the possibility that a bad drug is approved. And we can see that this is a fairly large portion. Okay, do we want a bad drug to be approved by this high possibility only to ensure that any good drug will be approved? Maybe not. Okay, or maybe yes. Depends on the situation, depends on whether the bad drug will make a lot of bad effect on human health, and depends on how beneficial of a good drug is. So for example, you don't want to miss any good drug because this good drug saved tons of millions of people's life. Okay, so then in that situation, maybe we'll put it in T1 star. Okay, let me see if I can erase it. Yes, okay. And then let's look at the other extreme, which is T5 star. Okay. So at T5 star, because T5 star, this line is completely at the right-hand side of the bad drugs distribution. So in this case, we are making sure that none of the bad drugs will be approved. The problem here is that we might miss a lot of good drugs, and the probability that we miss any good drug is on the left-hand side of this T5 star. Okay, so is this yellow area? Okay. All right, so what kind of situation would justify this decision to draw a line here? Then maybe any bad drug will cause severe harm to human body. So we don't want to approve any of them. At a cost that some good drugs might be rejected because maybe the good drug doesn't bring so much benefit to the society, but one bad drug would be so damaging. So that depends on the situation. You might think of, okay, then I would just draw it at T3 star. The answer is yes and no. We want to have a situation where we maximize the social welfare. So when we talk about good drug and bad drug, we have to think about what's the good drug's benefit and what's the bad drug's side effects. For example, in terms of a pandemic, if the pandemic is killing a lot of people, then we don't want to miss any good vaccines, right? So we want to lower our type 2 error, but we also don't want to bring in vaccines that are very harmful to people. So how harmful are a possible bad vaccine? So far, let's make an assumption, okay? I'm not an expert of that, but let's make an assumption that vaccines at maximum, it's gonna cause people to have a fever or some pain. And let's say some at maximum, it's gonna cause some blood clots in very vital areas of your human body. We wanna minimize that, and we wanna control that at a certain level. So then maybe we'll put it in T2 star. Okay, so that is a level that will be discussed by the FDA committee in a certain areas of experts. Okay, all right, so this plot, in conclusion, this plot talks about trade-off between type 1 and type 2 error. Regulators needs to balance social welfare and potential harms. More incentive to avoid type 1 errors because of media attention. This is particularly severe when some cases of side effects can be exaggerated when media, or sometimes when we say media that is not fact-checked, or media that doesn't understand statistics, can be exaggerating the harm. Type 2 errors rarely get in the media because it's very hard to catch. It is easy to catch that a bad drug is approved, but it's difficult to catch that a good drug is not approved. Okay. We have other regulations. For example, doctors have prescription power. This is true in most countries. The benefit is that there is less intentional and unintentional abuse of drugs. The cost is time, inconvenience, and expenses. There's bans on direct-to-consumer advertising. Bans in places in most developed countries except US. So in US, you can see a lot of advertisements about medicines, but in a lot of countries, they believe that this is misguiding consumers because consumers only know they want to feel better, but they might not know enough about a medicine merely from looking at your advertisements. Okay, so the benefit of that is to prevent moral hazard, reduce strain on doctor-patient relationships, while the cost is that consumers may not find out about new drugs that will benefit them. All right, so conclusion of this chapter is we have a lot of trade-offs for patents. We have trade-offs between incentive for innovation and affordable prescriptions. For government price controls, we have trade-offs between innovation and affordability. For FDA regulations, we have trade-offs between more new drugs and fewer dangerous drugs. Type 1 and type 2 errors are trade-offs. There's a trade-off between approved bad drugs and decline good drugs. Lastly, for doctor prescriptions, there is a trade-off between the increased safety of drug use versus expensive drugs.